So, good evening. My name is Ankita. I am a TA for the course on Basics of Biology by NPTEL. So, welcome to the live interaction session for week 7. Today, we will be discussing a few topics. Let me just share my screen for a moment. Okay, so so on the agenda for today, we will basically first be going over a recap of the topics that were covered in week 7. And then we'll go over the questions that were given in the assignment for this week. And finally, we'll go through some sample questions, which are a part of the topics that will be covered in week 8. So before we get to the assignment, let us first revise and just basically go over the topics that were discussed in week 7 once more. So till now, what we have already discussed from the start of the course till now, let's just go over that once. So first of all, the objective of the course is to look at living organisms, and we are trying to take a very holistic approach towards that. So with that in mind, first we understood how living organisms have evolved. How living organisms have evolved from the origin of life, how Earth has become more and more conducive for living organisms to survive, which is what we looked at in evolution. Then we understood cells, we understood the different types of organisms that reside on Earth, how they are different from what they initially were. We learned about biomolecules, individually carbohydrates, proteins, lipids, etc. Then we are currently understanding cellular processes. In the last live interaction session, we had looked at a few topics, and today we are going to go over a few more, which is understanding cellular processes part 2. So under this, we have the immune system. Sorry, so much. We will be looking at the immune system. We'll be looking at a few topics like vesicular transport and one or two other details as well. So first, understanding the immune system. So the defense mechanism of the human body, or rather of any living organisms, in this case we're talking about humans, is the immune system, right? So it is it has many subfolds as well. The details are not being taken up in this course. It's beyond the scope. But overall, if you look at the immune system, there are some barriers or some defense mechanisms which are physical in nature. Like, for example, your skin is the first major protecting mechanism that persists in any living being, whether it is for some animals, in our case it is our skin, the hair, etc. So these are all initial barriers against invasion by any other organism, in particular parasite or bacteria, virus, etc. Those are physical mechanisms. What we are currently looking at are the cellular mechanisms, which are, you can call them as internal mechanisms within the body, the mechanisms that are kept in place to make sure that in case these physical or anatomical barriers, they have failed, or not failed, if they have been sort of overtaken by these intruding organisms or invasive organisms, then what are the mechanisms that come into play? So that response to invasion by such a living organism, by such a invader or a pathogen or whatever sort of parasite has come in, is what you basically understand as an immune response. So the response of the host immune system to that organism is the immune response, right? Very broadly classifying, it is humoral and cell-mediated immunity. What does humoral mean? Humoral, it comes from the word humors, which basically means the liquid part, or you can say the fluidic part. On the other hand, cell-mediated, as the name indicates, obviously is centered around cells in the immune system, which are acting as checkpoints, or which are ensuring or which are maintaining good health in an individual under normal default circumstances. So here we see that humoral mediated immunity or humoral immunity, which is brief, like in abbreviated and called as HI. It involves antibodies. Why antibodies? They are not liquid, right, or fluidic in nature. Then why are we calling them? Because antibodies are usually circulating in the blood, in the sera of an individual. Serum or sera is the fluid part of the blood. So they are circulating in that, and hence they are included under the humoral immunity. And they are majorly involved in defensive mechanisms against pathogens and particularly viruses. On the other hand, cell-mediated immunity, very largely talking about it, is mediated by T cells and B cells. Like if you talk about cell-mediated immunity as a broad barrier, it can be very broadly classified into B cell and T cell mediated immunity. Interestingly enough, antibodies, what we understand as antibodies, are actually receptors present on the membrane of B cells. So B cell receptors, or BCRs, are basically antibodies. On the other hand, T cells are majorly helper T cells and cytotoxic T cells, which is TH and TC cells. These help the B cells. They are involved in a number of processes, but they also, among others, they are also involved in helping the B cells release these, first recognize a pathogen and then secrete or release antibodies which are specific for the pathogen that has come in. So these two arms, HI and CMI, are not completely independent of each other. They are interconnected, and either one is not always the best strategy against a pathogen. One of them always takes over, but they are effectively interconnected. So cell-mediated immunity, however, is majorly involved in fungal, viral, and intracellular bacterial pathogen protection. When we say intracellular, we mean pathogens which enter the cell, take over the host machinery, and effectively affect the living organism's health. And CMI is also the major defense mechanism that comes up against cancers. In fact, among the cell-mediated immunity also, there is a special class of cells which are known as natural killer cells, or NK cells, that take over this huge job. And for reference, if you are interested in immunology and want to understand, like, from the basics to quite a lot of detail, there is a book called as QB Immunology, which is like, you can consider it the God book. It has every detail, and if you want to understand any basic concept, it is 110% recommended to go through that book. It will clarify a lot of things for you. So for now, as part of this course, this is the information we are currently focused on. And after that, we are understanding antibodies. So antibodies are, like we said, they are part of the humoral immune system, right? So if you look at the structure, what we have are two heavy chains, two light chains, which are connected by disulfide linkages. Here are the disulfide bonds or disulfide linkages. Among them, these sites on the, like we also discussed in the previous live interaction session as well, sites on the antibody, like if you consider this as, if not, this is the antibody, and the site on the antibody which binds to the antigen is known as the paratope, and on the antigen, the site where the antibody will bind is known as the epitope. Apart from that, if you look at, like, the antigen binding site, here you can see it more clearly, and this is basically the it is the paratope. And other than that, you can see that each of the heavy and light chains are made up of little fragments, which you can see as variable and constant regions. The variable region is where you find as the name indicates, it's variable. It's because there is a huge diversity or a huge number of combination of so let me put it differently. The variable region is actually responsible for giving us, every individual, this immensely adaptive immune system. Without going into much detail, because there's a lot that goes in, there is a lot of recombination, etc., that happens to actually give you the variable region. What it does is it is variable. It can change based on what is the attacking entity, and it can provide defense against that. That is actually why the immune system is so all-encompassing, which means that it is able to protect us against a huge range of pathogens, mainly because the variable region can fluctuate or can change or vary a lot. The remaining portion, this C portions. These are constant, these are conserved, and they are required for other purposes. Now, how is an antibody produced? So production of an antibody, first obvious reason that you don't, without a reason, yes, antibodies can be produced, but the change or variation in the level of an antibody will only come about when there is a antigen which has come in, which is selectively increasing the level or the expression of one particular antibody. So if you consider 4 different steps, or 4 different phases or levels of expression, then what you can see is, first of all, you will find a lag phase where you will have a very basal or very low level of an antibody. Then what you will find is the log phase, which is where you can see a very steep increase. So at this, somewhere around this place, you will get an antigenic stimulus, which means there is a sudden increase in the level of an antibody because its cognate antigen has come in. So there is a requirement to eliminate that antigen, to eliminate the pathogen that has come in, and for that the immune system will end up increasing, suddenly spiking up the level of this antibody. So that is the log phase. Then comes the plateau phase, where the antibody synthesis and its use is kind of equalized to a large extent, which is where the expression it kind of plateaus off, which means it gets a little bit saturated. And then finally, there's a declining phase where you will find that there is a decrease in the antibody titer because whatever pathogen was circulating in the body at that point of time has been eliminated and it is no longer required. Then we come to the same process in a little more detail. So first is that the antigen is entering. If you consider this is the antigen, right? It has entered into a living system. Now, antigen presenting cells, which are APCs, commonly called as APCs, majorly include B cells, macrophages, and dendritic cells. These 3 cells are, as the name indicates, antigen presenting cell. What does it mean? It means that once an antigen has entered into a living system, what these will do is that they will take up that antigen, chew it up. Chew it up means that they will digest. So you have a large protein, some part of some bacteria, it maybe its cell surface receptor, a bacteria has been taken up, or some sort of a pathogen has been taken up. So macrophages, dendritic cells, and B cells, what they do is they completely engulf it. If you see here, it is going through the cell, right? In this case, the macrophage. It's completely engulfing that antigen, digesting it into small fragments or peptides, and it is presenting it on its surface via MHC. MHC is a class of receptors which are called as major histocompatibility antigens. So what they do is, via class 2 MHC molecules, they present the antigens on their surface. These antigens are then recognized by the humoral branch of immunity, by the cell-mediated branch of immunity, which are T cells. So T cells, and in particular, if you look at the picture, you'll be able to see TCRs. This is a helper T cell, and these are TCRs or T cell receptors, which are able to recognize the antigen. And there are some other co-receptors also which come in, which is a lot of detail for this stage of learning. And then they recognize that, oh, this is that antigen. And in response to that, they stimulate B cells, which are also a part of this humoral immunity, to selectively allow the activation of those B cells, which will be able to produce an antibody against this particular antigen, and they allow it to increase in number. And it undergoes a process known as clonal selection and clonal expansion. Selection, why? Because antigen A has come in, so we need an immune response against antigen A. So a B cell which is specific for antigen A will increase in number. It will get clonally selected, and in response to its selection, it will expand, which means it will keep on multiplying and producing plasma cells and memory cells, and ultimately it will give us antibodies from the plasma cells. So that is pretty much the cycle that an antibody goes through. Following that, we have polyclonal versus monoclonal antibodies. When antibodies are derived from multiple clones, they're polyclonal, majorly from animals. But if you need to synthesize monoclonal antibodies, when we say monoclonal, it means when an antibody is specific to only one epitope, which means that it is as specific as it can get, right? It is binding to one epitope on one antigen of one pathogen. So that makes it extremely specific. And as you can understand that the process of getting it is also extremely expensive. And therefore, some like monoclonal antibodies are used mainly either for medication purposes, or they are used in research only for some very crucial experiments, or experiments that do not require agglutination or processes like that. So it's a cautiously chosen reagent. And it is derived using a specific technology or process or experimental strategy, you can say, which is known as the hybridoma technology. And as you can see here, these are polyclonal because you have plasma cell 1, 2, 3, 4, 5, which are recognizing antigens 1, 2, 3, 4, 5 from the same, or which are recognizing epitope 1, 2, 3, 4, 5 from the same antigen. So they are responding to the same antigen, but to different parts of it. Compare that to monoclonal, then all of the antibodies are recognizing only epitope 6 from that antigen. That is a monoclonal population. So how will you generate the polyclonal antibody? First, you will have a hapten. A hapten is a very small small part of a protein which can elicit an immune response, and you prepare that antigen. You immunize animals. When you say immunize animals, it basically means you introduce this particular antigen in the animal, and in response to that antigen, you will have the organism will elicit an immune response, and you use that antibody, use those antibodies which are generated in the organism for your own purpose. So, like for example, we talked about ELISA, we talk about Western blotting, and so many other immunotechniques. In those cases, you need antibodies, right? All those experiments are heavily reliant on the immune supply. So there we use antibodies like anti-goat, anti-rabbit, etc. This depends upon the organism in which it was raised. So organisms like these are chosen mainly because of their closeness to humans, one of the reasons, and secondly because they are organisms which are able to produce antibodies at a large scale. You can get a lot of blood from them, and effectively also a larger volume of serum, and in response you can isolate a larger proportion of antibodies from such organisms. Now after that, you will test over a period of time. You will sample a little bit of blood from the organism and check that yes, antibody has been generated or not generated. And then after you will track how much of the antibody is being generated, and then after a definite period of time, the animal is euthanized, and from that animal you collect the serum, you purify antibodies, but you know that because you had introduced an entire antigen, like in the previous case we saw there were 6 different epitopes. In reality, in the real scenario, there are hundreds of such epitopes. So antibodies can be generated against each and every one of them, and they are specific for the same antigen but different epitopes. Therefore, the entire population of antibodies that you will get would be polyclonal in nature. So this is the process is described in detail here. We have first seen that for the antigen first needs to be purified. Then you have to prepare the immunogen. So antigen, along with, in this case, it's FCA, which is used, which is Freund's complete adjuvant. It is required when the antigen that you're using, when it is when its molecular weight is low. If it's a small antigen, then you need to combine it with some sort of an adjuvant like this to increase the molecular weight and effectively increase the immunogenicity of the immunogen. So there is an antigen, there is an immunogen. Every immunogen is an antigen, but not every antigen is an immunogen. This is a statement that needs to be understood with clarity. Antigen is any part of a protein or a peptide which may or may not elicit an immune response. And immunogen is most definitely a part of a protein or polypeptide which is capable of eliciting an immune response. So that is prepared, and for the process of immunization, you need a little bit of immune blood so that you can contrast it, and then you would carry out an injection of the or you would inject the antigen into the or immunogen into the animal, which can be intravenous, intraperitoneal, subcutaneous, or intramuscular. And after that, what you would do is you would determine the antibody titer at some definite time periods, and then you would check what is the level of the antibody using a method like indirect ELISA, and for that you would have to purify the sample. Just a second. So that completed lecture 1 for this week. Then in lecture 2, continuing with the same theme, we were introduced to the technique which we very briefly just mentioned its name, which is the hybridoma technology for monoclonal antibody production. So hybridoma, if you just think about the name, it means that a hybrid is formed, right, at some point of time. Now, what is this hybrid? This hybrid is actually a fusion or a hybrid of myeloma cells and B cells. So for that, sorry, for that, you need to have myeloma cells in culture, and you would fuse them with B cells that you obtain from an animal's spleen. You fuse these two using reagents like PEG, polyethylene glycol. There are other fusion agents as well, but most commonly PEG is used in the laboratory capacity. Now you have to check and verify that yes, the entire population that you are using in the subsequent steps, it is made up of purely fused cells, and then you harvest them. So here we have, next we have the antigen preparation, emulsification of antigen, immunization, testing antibody generation, bleeding, serum collection, and antibody testing, much like the process of polyclonal as well. So here you see a diagrammatic representation of the same. So these are myeloma cells. These are mice. So where are you getting these spleen cells? You will have a mouse model under most common circumstances, or you will have some sort of an animal in which you are introducing an antigen. Now, always keep in mind that when you are generating an antibody, most definitely your animal would not be immunocompromised. We use immunocompromised mice when we are trying to grow a tumor, or when we are trying to perform some sort of other experiment where we don't want the immune system to reject tumor growth or cell introduction or some sort of transplant or something. But when you want the antibody from the animal, most definitely you would want an immunocompetent mouse. And within this these mice, the antigen is introduced, and after a definite period of time, when we can expect that they are 4 to 6 or 8 weeks old, then you can or 4 to 8 weeks after injection, you can expect a persistent immune response to be present in them. Under those circumstances, you take the spleen cells from such animals, and these spleen cells are fused with the myeloma cells that we talked about using reagents like polyethylene glycol. Now, what you would get, if you look here, you will see that you get a mixed population. Some of them are fused, some of them are not fused. Even under the not fused population, you will have individual myeloma, individual spleen, and spleenomyeloma cells as well, like the combination of the two. What you selectively want is you want to grow only the hybrid cells from this entire mixed population. And what is the reason for this? The reason is that these are the cells you can expect to produce your desirable antibody. So you actually culture them in mass culture and you get the antibody from them. Or what you can do is, after you have propagated several clones of the fused cells, you can freeze thaw it and induce tumors in mice to get antibodies in new mice. So either of the two techniques can be opted. So if you look at the detailed protocol for this, then first of all, you would have to prepare the peritoneal execute cells. So PECs, or peritoneal execute cells, how are they obtained? From bovine hypothalamus to serve as the feeder cells from hybridoma for hybridoma culture. So first you would sacrifice non-immunized mice. You would dissect out their peritoneal cavity. You would use DMEM for this purpose, and you would use those cells and plate those cells on a culture dish, and you would incubate them for some time and check for contamination in like periodic durations. Then you would sacrifice the non-immunized mice either by, just a second, where is that? Yeah. So we were talking about the preparation or the methodology which is used for the hybridoma technology. So you would first have to prepare the spleen cells. For this, you sacrifice these mice, and you would next soak them in a beaker, like the dead mice are soaked in a beaker. Their abdominal regions are dissected. You remove the spleen into a petri dish or incubate the cells in a Gray's hemolytic solution. This hemolytic solution is made up of Gray's A and Gray's B in a 1:4 ratio, which is about 8 ml of Gray's A and 2 ml of Gray's B, and it removes RBCs from the spleen cells and only leaves behind myeloma cells, which is the material of interest. And the reason I'm like hovering over this part particularly is because it's one of the assignment questions as well. Then you collect the cells, plate them in a tea flask, and allow them to grow. And you resuspend them in serum-free DMAM at room temperature. Now, for mixed in a ratio of either 5:1 or 10:1, and then they are centrifuged, supernatant is removed, reduced polyethylene glycol, particularly here PEG 6000 has been mentioned, which is added to allow fusion to take place. So then they are resuspended and with FCS, and then these are added upon feeder layer of the PEG cells, which we had previously discussed about peritoneal exclude cells, and on top of this, you would add the fuse cells and incubate them for about 24 hours at 47 degrees 5% CO2 before you go for screening steps. So now you have B cells, you have you have myeloma cells, and now you are trying to select only for the hybrids. Now hybridoma cells, they have what is happening here? What is happening here is that you have cells from the mice spleen, right? Peritoneal exclude cells are the feeder cells, and apart from that, you have these spleen cells. They are capable of producing antibodies. Myeloma cells, myeloma is a type of cancer, right? You know that. So myeloma cells they are not producing antibodies. They are not immune cells, right? But because they are cancerous cells, what they have done is they have their replication or cell division system is compromised, and they are dividing at a or they're proliferating at a very abnormal and at a very accelerated rate, which is beneficial for us. Why beneficial? Because when you are culturing these, when after fusion, when you want to select for only those hybrid cells, you're culturing them in this sort of medium which have this fusion, which are carrying this fusion to proliferate, while other cells which are fused. On the other hand, if you look at the myeloma cells, they are not able to grow in the hat medium because there is a minopterin which is present, and these cells cannot offer. We'll get into details of this, and it is only allowing you to allowing us to culture the hybrid cells selectively. When we come to the molecular mechanism of this selection, what it actually depends upon is it depends upon cell division, which is reliant on normal nucleic acid or nucleotide biosynthesis pathways. Why? Because if you don't have an abundant acid pool, then such a cell is not able to replicate its DNA, and therefore such a cell is not able to proliferate, and it would eventually die multiplication. Now, how to allow those to survive? How does it inhibit the de novo pathway again? It has aminopterin, which stops the process of nucleotide biosynthesis, and de novo pathway therefore becomes blocked in such cells. And HGPRT enzyme, which is present in B cells, absent in myeloma cells, is present in the hybrid cells. This HGPRT enzyme takes over a backup or a salvage pathway, whereby it uses hypoxanthine, which is present in the medium, to produce guanine, and it allows only the hybrid cells to survive, myeloma cells, because they're HGPRT deficient. So only cells that have a functional salvage pathway would be able to survive, and these cells must have the HGPRT. Now, since myeloma cells entirely completely depend upon de novo biosynthesis, pathways. So when you have HAT in the medium, or when you're using the HAT medium, individual myeloma cells or kidney cells will not survive, but hybrid cells would be able to do so. So you you will culture such hybrid cells, and then after a requisite number of generations, you would isolate the cells by serially diluting them. You would get single clones from them. You would delineate the boundary. You would remove their media, trypsinize each colony, wash it, transfer it to a 24-well dish, allow them to become completely confluent. You would expect an entire population of that exact same cell, and then you can passage it further and further to increase the cell number. And then you must certainly check the expression of the antibody of your interest using ELISA. That is like the entire elaborate process that we have actually to look for that same, right? See that the medium changes color, which is also something we have previously discussed because of the presence of phenol red-like indicators. Then you change the medium, and you expect that healthy cell lines would produce antibodies, which would be passaged for more and more generations to get more and more antibody titers. And then you harvest the supernatant by transferring it to a tube, centrifuging it, transfer it to a fresh tube, adjust add sodium azide and preserve the supernatant. Why sodium azide? Sodium azide actually helps in. So if what happens is that if you let an antibody, if you just harvest antibodies and keep it as such, then within a week or so, you can find some clumping which occurs. Some clumping which occurs, and rest here is that how are you at all purifying these antibodies, and that is done using columns. You can do so using equilibration, using columns. You can do so starting off with equilibration of the column that you're using. Prepare the sample, wash it a few times with an equilibration buffer, elute, and you regenerate the column again. So here you can see, if you look at the time of elution and you plot it against the absorbance, then you will find very minimal absorbance initially when the sample is injected, and after that you would carry out equilibration or washing. Then against the sample, you would find a sudden spike or sudden increase in the absorbance because of the antibody which is clonally selected and expanded as well. And after that, you would elute and you would find a reduction in it in the antibody titer, and obviously the corresponding absorbance after a definite period of time. Now, antibody and antigen are capable of forming cognate pairs, which means that they are, you can think of them as two halves of the same whole. So they are completing each other in a way. This is also indicating the same thing. Now, antigen can either be insoluble or soluble. If it is insoluble, it would agglutinate. If it is soluble, which means that if it remains suspended in water, does not separate out, not water in the solution. If it does not separate out, then it is said to be soluble. And the process that is taken up under those circumstances includes precipitation, radial diffusion, and immunoprecipitation, which can be used to study it. Under immunoassays, we have ELISA, Western blotting, which we have already talked about. Western blotting is also called immunoblotting, and RIA, or radio immunoprecipitation assay. Now, we talked about agglutination for insoluble antigens. So agglutination is the process where several that it forms very large aggregates, very large, very visible aggregates are formed. So it is mainly centered around particulate antigens like bacteria, virus, or RBCs. Very sensitive, very reproducible process, and agglutination can either be direct or indirect. When it is direct, what it does is it's a test which diagnoses antibodies against a large number of antigens like RBCs, bacteria, and fungi. And you have here you have antigen antibody mat which is formed, which is present at the bottom of the cell. And if either of the two is not present, then agglutination would not occur. And insufficient antibodies are present as a result of that mat would not be formed. So here you have antigen in the figure, you have antigen. What is happening here is that they are sort of linking together. Now, this agglutination reaction, as dooming as it may sound, also has a few positive aspects to it as well. So one of the major utilities of this concept was brought about when we started understanding how blood group is typed. So it basically looks at four major blood groups, which are Karl-Landsteiner, and Landsteiner's strategy was centered around four different blood groups: A, B, AB, and O. So you can see that individuals with each of these blood groups have a difference in the antibodies that are present and the capacity for agglutination in them. So blood types are they take into consideration both the ABO antigens and the RH antigens for each case. On the other hand, for indirect agglutination, your diagnostics as well, particularly when you're looking for some soluble antigen, for example, streptococcus. So if they get absorbed into particles, onto particles, soluble antigens are capable of responding. So antibody reacts with the soluble antigen that adheres to these particles, and therefore they agglutinate in a similar manner to direct agglutination tests. So that completes the immune system concept for this week. And next we'll come to the process of phagocytosis in just 2 minutes. So, yeah, so we were talking about third lecture, and in the third lecture, we'll actually try to wrap up the remaining pictures about phagocytosis. So initially, you can see the materials and all have been discussed. Basically, phagocytosis, as he had explained, is the process of taking up some material into the cell. Phagocytosing means you are eating something up, you are taking something up into the cell. So for this process, what he had discussed was the method via which you can actually study the process of phagocytosis in quite some extensive detail, where you culture this particular cells, and you use you actually use latex beads which are taken up into you actually beads are taken up to those cells to monitor how phagocytosis is occurring. So real-time study of phagocytosis is done you'll see here, we'll be able to see here by fluorescence microscope as well. What is usually done to actually understand phagocytosis, the more acceptable, um, more accurate, even you can say, more accurate, more acceptable technique is to use a flow cytometric phagocytosis assay, and we'll get into details of that directly. Here you just have the setup, the principle, and the data, the kind of data that is generated. So in this process, first what you do is you want to identify the phagosome. The phagosome, as the name indicates, it is the structure which um, okay. So for some reason the recording got interrupted, but just a second. We were talking about phagosome uh, the experimenting, and then plate around 10,000 cells on cover glasses and incubate them inside 24 well dishes. After that, you produce a cocktail of FBS and antibiotic, and then incubate cells overnight, allow them to attach to the cover slips. After this, you wash the cells with DMEM that does not contain FBS. You don't want any additional growth factors to be there. Then you prepare the suspension of latex beads without this FBS. Again, this is serum free. This is also commonly called as the serum free media. And then the media is removed, and bead suspension is added, and the cells are centrifuged in 24 well dishes. Following this, the phagosome is identified by incubating the plate for an hour, and it is washed well with DMEM to remove any beads that have not been taken up. Uninternalized beads are removed, and then the biological sample is fixed and hydrated, following which they are stained with philippine, which is a specific stain, and then you use mounting medium, fix the cover slip, and then you study it. Now, to label the lysosome, what is done is that their cells are cultured on cover slips, and they are grown with rhodamine dextrin, which with this specific medium containing antibiotics, and they are washed and chased for 1 hour in a medium which does not contain sorry. So cells are observed in bright field, and if the bead has a blue fluorescence, then the cells can be visualized through the red channel. And this is basically the method that is used for study. In the fourth lecture for this week, what was discussed was majorly centered around apoptosis and flow cytometry particularly. So here you can see that different organs have different types of cells, like blood, liver, brain, and heart are all made up of their heterogeneous mixture of different types of cells are present. So if we look at the cellular properties, then each cell has difference in density, size, metabolic reactions, and cell surface receptors. In particular, you can see that for mammalian cells where there are sperms, RBCs, etc., each having different sort of dimensions. Now, why are we suddenly talking about dimensions? That is because when you want to separate or segregate or sort cells in different aspects, what you look, one of the ways to actually do that is to look at their size. And the second aspect, which we will be discussing pretty soon, is to look for their granularity or intracellular density or composition, you can see. So here we are looking at a cell analyzer where you can see that if this is a laser beam and you have a flow of cells that is going through, what the detector can look at is that it can see what is the size of the cell and what is inside the cell. Based on these minute differences, it can actually separate them. Cell sorter, cell analyzer are used for this purpose, which is what you see in this instrumentation as well, which is what is present inside a flow cytometer or flow cytometry assisted cell sorting or fax machine. So this is a complicated setup, not going into it at the moment, but what it majorly has is a fluidic system, the flow system, which is generating a steady stream of the of cells, ensuring that it is going through through the nozzle in a manner such that you get one single cell at a time. You're not getting doublets, you're getting singlets, and each cell is getting separately, accurately analyzed. The second is an optical system, which is the reason why when a cell is passing through, the difference in its fluorescence, the difference in what the optical differences that are generated because of the difference in the cells is part of the optical system. And thirdly, we have the electronic system, which is processing whatever signal has come in. It is processing that, and that is the third aspect of it. So under fluidics, what we have is a flow cell where there's a port, a channel or a port through which you are introducing or injecting the sample. You have an outer sheath which contains faster flowing fluid. Sorry. So, sorry, I had to attend a call. So, the flow cell and hydrodynamic focusing. Hydrodynamic focusing is the process, which is what I was just describing previously, which is where you are ensuring that a single continuous stream of fluid is passing through the laser where there are individual single cells, not doublet, not clumps, but single cell and a steady stream, uninterrupted steady stream of the cell suspension is flowing through. Then comes the flow cytometry optical system, which is where you have a laser and you have an arc lamp. When we talk about lasers, there are argon lasers majorly, which have a blue to blue green range in the 488 nanometer wavelength. And we have detectors, which involve a forward scatter and a side scatter channel, which is what we were previously discussing, that cell analyzer or a cell sorter will majorly look at two different aspects. One is where it is looking at the cell size, the it is looking at, and basically it's studying the refractive index of the cell as well. And the intensity also depends upon the cytosolic structure, like I just said. And thirdly, the electronic system, it mainly converts photons to photoelectrons, measures the amplitude, area, and width of the photoelectronic pulse, and it amplifies pulse either linearly or logarithmically. Now, the different types of plots that are generated are single color histograms. This has been discussed. I'm not getting into detail. Now, the data that you get, all of the data that you get, it will be quite diverse because the population can be quite distinct. So there is a particular strategy known as gating, where you narrow down or you sort of place a boundary on the cells that you are going to be looking at. And what it does is it gives you gives you a subpopulation of cells that you are particularly interested in and that you can analyze with much greater detail and much greater accuracy circumstances. So you're looking at rectangular, elliptical, polygon, different shapes of plates can actually be used, and that has been also demonstrated as well. So these are different types of dyes which are used for us for this process of flow cytometry, like FITC, fluorescein isothiocyanate, 5-query-3 indexes red, and so on and so forth. So what is the purpose? Why did we come to it? Because that's majorly flow cytometry is used for cell cycle analysis, to study apoptosis, and for sorting as well. So here you are seeing that cells can die majorly for via two processes. One is known as apoptosis, which is programmed cell death, where a cell is consciously taking the decision that it is now no longer going to operate survival strategies. Instead, it is going to proceed towards death. And necrosis is a more aggressive, uncoordinated, unprogrammed strategy where a cell sort of bursts and it swells up, bursts, and then dies. So this is the experimental design where we're first understanding how trypan blue works. It's a charged dye. Living cells do not take it up because there's a membrane potential which is present. Dead cells, they take it up, and you can actually use that for cell counting. Then under the procedure for studying this, you will first have to remove cells, mix it with trypan blue, fill the hemocytometer chamber, observe it under the microscope. Viable cells are colorless, dead cells are blue or colored or dark in color. You will mark them, and nowadays, in fact, there's a more automated process which is present, like Countess is one example of such an and present in your population. Talking about cell cycle, this is how flow cytometry can be used to study what cells are in what is the percentage of cells in each phase of the cell cycle, and also what are the different apoptotic strategies that are taken up. And majorly two things are studied when you are trying to see if a cell is dying under like when you're trying to solve a research problem and you're trying to see what is the effect of the treatment or the strategy that you have taken up, and you want to see if the cell is going to die. Then under those circumstances, you will either analyze the condition of the cell's DNA, if it is broken, if it is undergoing some sort of damage or something like that, or if there is the exposure of phosphatidylserine, which we have previously discussed to be an eat me signal, normally present in the inner leaflet of the plasma membrane. It is brought to the outer leaflet, signaling that the cell is going to, it's going to die. Sorry. And in the last lecture, the topic that was discussed was vesicular transport. So as the name indicates, it is the entire strategy which is used by a cell to transport uh, proteins that have been synthesized, or you can also call it cellular cargo, from one place to another using vesicles. It's a trafficking system that is present inside the cell. Now, what are the organelles that are present? It is the endoplasmic reticulum, the Golgi, the plasma membrane, lysosome, all of these. Majorly, ER, Golgi, and lysosome are the three major players in this entire extremely well-regulated process. So that is what they're doing. Now the process, it can start off with the taking up of solid material from the outside, which is the process of endocytosis. So when we talk about proteins, that is the protein sorting process that we have already discussed. Here, when we are talking about vesicular transport, it revolves around taking up some resource from the outside, or some material is taken up from the outside. So that is, it starts off with endocytosis when it is a solid material. When it is a liquid, then it is known as pinocytosis, and it delivers the protein to different organelles. So material present outside the cell binds to the cell surface through the receptor. It gets trapped in an endosome. Endosome goes to late endosome, lysosome, phagolysosome, etc., and then finally it goes to Golgi and then gets distributed. So we are looking first at the ER. It is smooth and it can be rough or smooth depending upon whether ribosomes are present or not, which is what gives it that appearance as well. And protein synthesis on ribosome it is attached to the RER, gets sorted into three categories, either as integral membrane proteins, proteins for secretion, and proteins destined for different organelles as well. So if they have a signal peptide attached to it, then that gets recognized by signal recognition particles. Sorry. Signal recognition particle, and it gets delivered to its accurate destination. So under ER, these are its different major different functions, including steroid hormone synthesis, detoxification, sequestering calcium, protein sorting, protein modification, etc. Golgi bodies, first visualized by Camillo Golgi. We have talked about cis, medial, and trans phases of the Golgi. It's a network. It's also often called as Golgi network because there are multiple cisternae which are arranged one on top of the other like a stack, like a network which is present. But major functions of the Golgi include A, protein sorting, certainly. Second, glycosylation. Its huge contribution to the process of glycosylation is certainly something which is unique to it only, and it also is involved in proteolysis as well. Coming to the third organelle, which is the lysosome, discovered by Dewey. Very commonly, if you just think about lysosome, they're called as suicide bags of the cell, but that's not their only function. Majorly, this name comes about because of their role in autophagy, which is sort of a two-faced strategy taken up by a cell where it decides to die because it is understanding that the current availability of resources, the resources which are currently available to that particular cell, are not enough for it to actually survive. So it chooses to die. So the lysosomal lumen is extremely acidic. It contains proteases and other lytic enzymes that degrade the ingested material, and it can degrade pathogenic bacteria, it can degrade old proteins, etc. So glycosylation is a part of, so it's a trafficking signal as well, and it is a part of the biosynthetic secretory pathway that operates. And most proteins, approximately 50% of them, of all the proteins synthesized, go through this process, go through this alteration. And soluble membrane-bound proteins expressed in the ER undergo glycosylation, including all secreted proteins, surface receptors, and ligands. And thirdly, the types of glycosylation, NOC and phosphoglycosylation, are also explored. Among them, N-glycosylation is the most common. The process, it actually, or the nomenclature, it comes from where the glycosyl group is attached or the carbohydrate is attached. If it is attached to asparagine, which is ASN, or the one letter code for asparagine is N, then it is called N-linked glycosylation. If it is serine linked, then it is called O-linked glycosylation. So it's the most common type. Approximately 90% of the cases, as soon as a protein is synthesized, almost immediately and almost simultaneously with the translation process, this occurs. Then first there is assembly of the precursor glycan, then that glycan moiety is entirely shifted. You can see it here. You can see it here, where you can see that this is the entire glycan which is being synthesized. It is flipped onto the inner side, and then it is attached to the currently being synthesized protein, and then it is now glycosylated, and it acts as a trafficking signal. It undergoes further trimming in the endoplasmic reticulum, yes. It undergoes maturation processes which commits it for further transport in the Golgi, which is what we also described as one of its major functions, and that is the most commonly used strategy. O-glycosylation is more frequently or more commonly seen for high molecular weight proteins found in mucus secretions, proteoglycan core proteins forming ECM. Also found in case of antibodies. If you remember the constant region that we talked about, it usually has glycosylation, and that glycosylation is necessary for the antibody to remain soluble in the serum and not get agglutinated. And that is what we talked about. Then comes phosphoglycosylation, C-glycosylation, C-mannosylation, which is occurring by carbon-carbon bonding. And phosphoglycosylation is majorly limited to parasites like Leishmania and Trypanosoma, cymoles like Teostylum, etc. And this is the summation of the entire vesicular trafficking pathway. So that completes our quite extensive revision for this week. And yeah. We will go through the assignments and a few sample problems, and then we'll conclude the interaction session for today. So the first question asks us about the antigen binding site in an antibody. What is it composed of? So we have already discussed this, and you can look at the diagram shown here as well, where you would see that you would see that these are the heavy chains and these are the light chains. These are the hinges that we have already talked about, and these are the antigen binding sites. They are formed by the variable regions of the heavy chains and the light chains, VH and VL regions together, which gives the huge diversity to the immune system, which is also something we talked about previously. So yeah, so antibodies are secreted by macrophages, plasma cells, T cells, or dendritic cells. So if you remember, antibodies are basically B cell receptors, right? So we talked about antigen presenting cells. Antigens can be presented by 3 groups of cells, or APCs majorly include the dendritic cells, macrophages, and B cells. That is, these 3 are included under the same thing. But antibody secretion particularly is only carried out by B cells, and even under that, a specific subclass of B cells. So when a B cell has undergone that process of clonal selection and clonal expansion, what it is going to do is it is going to synthesize or it is going to produce multiple copies, and that includes plasma cells and memory cells. So memory cells only serve, as the name indicates, they serve the purpose of circulating in order to serve as a future preventive strategy in case cells are somehow getting infected by the same pathogen again. Then it would allow such an individual to have a stronger defense mechanism. A more rapid and a more heightened immune response could then be elicited under those circumstances. On the other hand, plasma cells are majorly only responsible for releasing or for secreting antibodies in very high titers, thereby acting as the defensive strategy of the immune system. Then comes the hybridoma technology. So in the hybridoma technology, B cells are fused with in the hybridoma technology, B cells are fused with T cells, plasma cells, myeloma cells, or mast cells. So among them, if you remember, these were hybrids made up of myeloma cells and B cells. And we used these two because of their unique abilities to, or because of the differential survival in HAT medium, whereby we understood that myeloma cells are immortalized, so they would be able to survive indefinitely, which is not something B cells can do because they're mortal cells. But on the other hand, myeloma cells will not be able to, do not have an operational salvage pathway, while B cells are able to do so. So because of their unique abilities or unique properties, these two cells are fused, and their hybrids are used for generation of monoclonal antibodies using the HAT medium. In question 4, we were asked that the blood from a person with O positive blood group will show agglutination with A, anti-A, B, anti-B, C, both anti-A and anti-B, or D, anti-D. The answer to this, since the question is saying that you are using a person who is O positive, right? O positive individuals do not have A or B antigen on their RBCs, but because it is O positive, it means that they are Rh, they're positive for the Rh antigen, but do not have the A or B antigens on the RBC membrane. So as a result, what we are seeing here is the what we would see here is that they would not be able to form or would not be agglutinating with individuals, would not be agglutinating with either A or B or with both. Therefore, by default, the answer comes to anti-B, but otherwise also you'd be able to say so because they are O positive in nature. Then comes the medium, which is used for selection of hybridoma cells, is LB, HAT, SD, or blood agar. We have discussed this time and again in different contexts, but yes, the medium is the HAT medium, which is hypoxanthine aminopterin thymidine medium, which is extremely selective and a brilliant strategy for generating and selecting such hybrid cells. Then comes the next question, that gray solution is used for removing A, PECs or PECs, B, B cells, C, myeloma cells, or D, RBCs. So if you remember, PEC cells are peritoneal exclude cells, which were from where we got the feeders for culturing. B cells are not removed, myeloma cells are not removed. In fact, RBCs are removed. Like when your objective is to remove RBCs, you would use Grays A, Grays B solution in the 4:1 ratio, as was previously mentioned in the lecture as well. And yes, it's basically a hemolytic solution. When we say hemolytic, basically, as the name indicates, hemolysis means RBC lysis or blood cell lysis, red blood cell lysis, heme containing cell lysis. So that is what happens there. In question 7, we were asked that what does aminopterin do? So aminopterin in the HAT medium inhibits de novo synthesis of A, nucleotides, B, lipids, C, amino acids, or D, sugars. So we have, so in this question, we would understand that nucleotides are obviously getting, nucleotide biosynthesis is obviously getting inhibited, like we saw initially also, which is why in the HAT medium, because of the presence of aminopterin, only cells that have a functional salvage pathway, or cells that are able to utilize aminopterin and which are HGPRT positive, would be able to opt for or would be able to survive and grow in such a medium. So that is the reason why aminopterin is used in the HAT medium, and it inhibits the de novo synthesis of nucleotides in this process. And since nucleotide synthesis is obviously an extremely survival essential process, therefore it is used for hybrid selection in the hybridoma technology. In question 8, we were asked that pinocytosis is the process of internalizing A, bacteria, B, virus, C, antibodies, or D, fluids. So the process of taking up or internalizing fluids is basically the process of pinocytosis. So you are taking up fluids, then you would call it pinocytosis. Question 9 is for Cell permeabilization, what is it routinely performed with? A. Glycerol, B. Glutaraldehyde, C. Tritinex 100, or D. Paraformaldehyde. Now, don't get confused. Paraformaldehyde is used for cell fixation, but when we are talking about just permeabilizing cells, so that if you are trying to perform, say, for example, immunofluorescence, and you need to introduce antibodies into the cell, then you would need to permeabilize it, or you would need to generate like holes in the cell membrane or gaps in the cell membrane. So that is done using Tritinex 100. So it's a non-ionic detergent, and it is used like the name, like detergent obviously indicates it. It is used in cell permeabilization, and uh, it does generate pores in the cell membrane, and therefore it is routinely used in the lab condition as well. Then in question number 10, we were asked that the site scatter channel, or SSC, it detects A, cell size, B, cell granularity, C, cell membrane permeability, or D, cell shape. Now we know that forward scatter, or FSC, is used to detect cell size, or it analyzes cells based on their size, based on their dimensions. Whereas the site scatter, because of obviously it means that if you have a cell and there is an incident light on it, then what is the change in the direction of that light is basically its scatter, right? So the site scatter which is generated is a result of the intracellular or cytosolic structure, or density that differs in such cells, and therefore the cell granularity would be the contributing factor under those circumstances. Then in question 11, we were asked that cytochrome C, which is released during apoptosis, where is it released from, right? So A is Golgi bodies, B from the nucleus, C mitochondria, or D endoplasmic reticulum, or ER. So the answer to this is the mitochondria. Cytochrome C is released from the mitochondria, and that is what we see here. Then in question 12, we were asked that calcium sequestration mainly is performed by Golgi bodies, lysosomes, endolysosomes, or endoplasmic reticulum. So like you can see the answer here, and it was mentioned directly in the lecture notes as well, that the ER, it serves as a storehouse for calcium. That is what we see there as well. The reason why you at all need to have a storehouse for calcium is because calcium serves as a secondary messenger, intracellular secondary messenger. So it is one of the most extensively used strategies for signaling inside the cell. Now, the level of calcium inside the cell actually dictates a lot of downstream processes. So under normal circumstances, you should not have free calcium circulating inside the cell, and therefore it needs to be, when we say sequestered, it means it is stored, controlled and stored somewhere. So that is why sequestration is extremely relevant and extremely crucial process that is taken up by the ER, and it is stored there at very high concentration, so that when required, there can be a sudden release of the entire, or there can be a steep release of the calcium from the endoplasmic reticulum into the cytosol, and then it would dictate whatever downstream processes need to be taken care of. And it's a very, very relevant process, and mainly just performed by the endoplasmic reticulum. In question 13, we were asked that in Golgi bodies, proteins are covalently linked. Linked with the sugar in the cis medial transphase or in the ribosome? The answer, it's a very direct question. It is done in the medial Golgi. Then in question 14, we were asked that glycosylation is the process of A, addition of an amino acid to a growing peptide, B, removal of a sugar from a protein, C, it is the addition of a sugar to a protein, or D, it is the addition of ubiquitin to a protein. So glycosylation, it is where you add a sugar, or maybe you can even say a carbohydrate, a carb molecule is added to a protein, basically. Then in question 15, we were asked that most common type of glycosylation, if you remember in the final few slides we had seen that almost 90% of all glycosylation processes are usually from arginine, usually from asparagine or N, which is why N-linked glycosylation is the most common. Yeah. Then we are going to look at some sample questions for the topics which are covered in week 8, and we'll just do so in a minute. So we'll look at a few sample problems now, including the topics of week 8. So since week 8 is majorly looking at topics that are centered around the process of digestion, the questions are somewhere related to that only. So the first question is about the type of nutrition where organisms take in food that is digested by enzymes which get secreted outside the body is A, parasitic nutrition, B, saprophytic nutrition, so C, holozoic, or D, autotrophic nutrition. So if you read the question carefully, you would understand that since the material being digested is something that is, or maybe you can look at it this way, that you are digesting whatever food material it is getting digested outside the body. That is only the indicative um, um, word or terminology. So that would be saprophytic. Saprophytic organisms, they majorly they rely upon dead or decaying and dying organic matter. So they actually release extracellular enzymes which go ahead and digest such material, and then whatever nutrients can be derived, then the other organism or the saprophyte, it takes it up. Parasites obviously are reliant on the host, and holozoic and autotrophic also do not fit into these criteria. The second question is about an internal parasite which multiplies inside the host cell, and it is life-threatening is A, Anopheles mosquito, B, fleas, C, housefly, or D, plasmodium. So if you look at the image on the right, you'll be able to see Plasmodium falciparum or Plasmodium malariae. As the name indicates, these are parasites or pathogens which cause extremely life-threatening and extremely crucial and very deadly diseases like malaria, etc. So they replicate in they enter the RBC and they enter such living cells, host cells, and they completely take over the machinery, hijack it, and such cells are obviously not able to survive. They lyse RBCs, and it's a very, therefore, a very deadly disease. And hence the answer is Plasmodium. Then in question 3, the mechanism of digestion in humans follows the order A. Okay, I'm not reading out all of the options, but then if you basically look at the subparts of each option, it is referring to ingestion, breakdown of food by mechanical enzymatic strategies, ingestion, absorption, and waste removal. So if you look at all of the options, obviously waste removal comes last, and very logically speaking, ingestion would have to come first. So the correct option would be A, ingestion. You first eat the food, you break it down because it's a complex substance. You can't digest it just as it is. So it has to be broken down, and for that there are mechanical processes which come in, which churns the food and breaks it down into smaller pieces. And there are enzymes, like for example, which will break polypeptides into individual amino acids, and it'll digest each component into smaller parts so that the stomach and the entire digestive system is able to take it up. It's able to break down the nutrients in a manner which can be utilized by that living system, and that utilization is basically the process absorption, which is sub step 3. And after absorption, not everything that you eat, it gets absorbed, right? After all of the nutrients have been taken up, anything that is toxic, anything that is not required is digested, it's removed, and it is excreted out of the system, micturition or excretion. Then in question 4, we are being asked that the slit-like space between cheeks and teeth is A, gullet, B, palate, C, vestibule, or D, buccal cavity. So just for reference, I have given an image here. So this is your cheek, and the teeth are present in like the palate is present like this, or here it's present like this, and you want to understand the space between the two, then these are known as vestibules, and there is one upper and one lower vestibule, and that is basically the answer to this question. Then in question 5, we have been asked that the involuntary muscle contraction that occurs in the entire esophagus or the food pipe for downward movement of food, or you might even refer to it as bolus, is controlled by A, okay, we'll come back to it. A, peristalsis, B, the spinal cord, C, the cerebrum, and D, medulla oblongata. So if you are not somebody who reads questions thoroughly, this would land you in trouble, because if you just read that the involuntary muscle contraction, you would immediately think of, oh, it's peristalsis, which is like the squeezing movement which pushes food down the canal, alimentary canal. That is peristalsis. But this question is asking you about the part of the human body. It's not even mentioned here that they're asking for some part of the brain, because you might just skip it somehow. So you have to be careful that what part of the body actually controls that this peristaltic process would occur. So that is actually done by the medulla oblongata part of the hindbrain. So be a little careful when you're reading questions like this, because these are intended to trick students, and it's very easy to get a simple question like this wrong, even if you are the correct answer. Then in question 6, we have been asked that the type of teeth which are used for chewing bubblegum are A, incisors, B, molars, C, premolars, or D, canine. Now, surprisingly enough, there are two answers to this. So if you talk about bubblegum, you never chew it with like your incisors or your canines, right? Because it requires a broader surface area, which is usually present in the teeth at the back. So if you look at the layout of the mouth, okay, let's just take the bottom teeth. Here you have the molars. the premolars, the canines, and the incisors. Sorry. So this is majorly taken up by the molars and the premolars. The center of the tooth that contains nerves and blood vessels is A. Cementum, D. Dentin, C. Enamel, or D. Pulp. So if you remember the cross section or study the cross section of a tooth, we'll get into this in more details in the next live interaction session on next Monday. So the center part, which is actually innervated, which is why because the outer part is obviously a tough coating which has to be durable, which has to be able to withstand all of the mechanical stress, ensuring that you put it through, and it has to be able to chew all of the food in order to be in order for it be digestible. But what is at the center is the innervated and the part which has blood flow, and that is actually the pulp. Then in question 8, we are being asked about the protective coating that ensures that the stomach wall does not get damaged by HCl is A. Mucus, B. Villi, C. Squamous epithelium, or a D. Just the epithelium in general. So yeah, so we are being asked that what is the inner coating on the wall of the stomach, which protects it from the acidic nature of the stomach acids. So that is most certainly the mucus, which protects. It's not just in the stomach wall; it's present along the alimentary canal in different portions of the alimentary canal as well, and it has several different functions. It hosts some of the gut microbiota also, which are required, which are necessary for breaking down a lot of food products or a lot of materials from the food that we consume as well. So it is, apart from that, also serving as a protective barrier or a coating for the stomach also. So it's the mucus, most certainly, yes. Then in question 9, we're being asked that the longest part of the alimentary tract is A. large intestine, B. small intestine, C. duodenum, or D. jejunum. So if you remember, basically the alimentary tract, it starts from your mouth, from your buccal cavity, and it goes up till the colon, right, till the rectum. So among them, the longest part is most certainly the small intestine, which has the maximum length, which is packaged into a very small space, and it is required so that inadequately done. It is not inadequately done, and it is slowly, steadily carried out in the small intestine, and therefore it forms the longest part of the alimentary tract or alimentary canal. Then in question 10, we are being asked that the backflow of fecal contents from the colon to the ileum is prevented by C. ileocecal valve, or D. the venous valve. So if so to maintain the flow or the directionality within the entire alimentary canal is extremely crucial, and these are maintained by some muscles, some muscular checkpoints, you may call them, which are known as sphincters. So there are which operate from esophagus to the stomach. There is a sphincter, so that stomach contents do not, under normal circumstances, do not flow back into the alimentary tract. Just a second, I keep getting a call. So to prevent such backflow, if there are pyloric sphincters and many other sphincters which are present, in this particular case, we are being asked that you don't want fecal contents to go from the colon to the ileum, like back to the intestine. And for that, we have the ileocecal valve. So the name itself is quite clear about it as well. You can see that there's ileum and the cecum. So from the flow is prevented in those directions. In question 11, we are being asked that the correct order of the large intestine is, yeah, it is, okay, I'm just going over the options. There is, you can actually see it in the figure here. It starts with the ascending colon, the transverse colon, and the descending colon, followed by sigmoid because it is sort of S-shaped, and then the appendix or the, you can avoid the, sorry, the appendix is a part of the ascending colon. From the sigmoid colon, we have the rectum, and then finally the anus. The anus is not shown in the figure here, but then yeah, that is the final part. So if we look at the sequence, then option B satisfies it. So we have ascending, transverse, descending, sigmoid, rectum, and anus. In question 12, we are being asked that the RNA and DNA are broken down into nitrogenous bases and sugars by nucleases in. So you basically want to understand where exactly is the nucleotide breakdown, acid breakdown being carried out. So this entire digestion process actually occurs in the small intestine. That's like very direct answer to this question, but yes, in the small intestine. In question 13, we are being asked that the cells that secrete intestinal antimicrobial proteins and also lysozyme is A, Paneth cells, B, goblet cells, C, enteroendocrine cells, or D, Argentafin cells. So your base, it's also a very direct question. You just have to remember what cells are responsible for what functions. So Paneth cells are actually responsible for AMP or antimicrobial secretion as also. In question 14, we are being asked that B, liver, C, small intestine, or D, large intestine. Then carbohydrate breakdown or breakdown followed by storage in the form. Then in question 15, we are being asked that cholesterol esterase and phospholipase enzymes are produced by A, tyalin, B, mucin, C, pancreas, and D, liver. So these are enzymes that are very obviously produced by the pancreas. So yeah, phospholipase and cholesterol esterase are all pancreatic enzymes basically. Coming to question 16, the disease that is caused by severe vitamin D deficiency is A, marasmus, B, kwashiorkor, C, constipation, or D, osteomalacia. So osteomalacia is the disease that is usually, if you don't remember much in details, then you can always keep in mind that vitamin D deficiency leads to bone defects or bone weakness, basically. So osteomalacia is caused as a result of it. Then in 17, we are being asked that the buildup of which waste material in the blood leads to jaundice. Also, if you've ever had a family member who suffers from this, you would have most certainly heard of it. So bilirubin is the enzyme which is like not the enzyme, the exact component that is usually attributed to jaundice because it leads to a lot of yellowing of such individuals. So that is what we can see here. Then in 18, we are being asked that the walls of the small intestine, small intestine consist of finger-like projections called as A, pseudopodia, B, villi, C, cristae, or D, fimbriae. So in the small intestine, you can find a lot of apart from a lot of infoldings, which are actually responsible for compacting such a huge elongated organ into such a compact or tiny space. Apart from that, if you think about the function of the small intestine, it is basically carrying out absorption, nutrient absorption, right? So you want to increase the surface area as much as possible. Sorry, as much as possible. And for that reason, we usually, you would require some extra support, some extra surface area from which absorption can take place. Now, in case of the small intestine, this is carried out by you can think of it as hair-like structures or finger-like projections, which are present throughout the small intestine, and those are commonly known as villi. So we have, if you look at the other options, pseudopodia, cristae, and fibrae, those are different. Pseudopodia is a different structure. It's usually present in individual cells, where those cells, they produce, or where they extend parts of their body to engulf, to phagocytose some sort of food material. Villi is the correct answer to this. Cristae you would find in mitochondria, and fibrae also are not the answer to this. Then these are the last two questions for the day. So in question number 19, we are being asked that the movement of Cl- or chloride during co-transport of glucose into the intestinal epithelial cells during absorption is by A, diffusion, B, osmosis, C, passive transport, or D, active transport. So during the process of absorption, when you are carrying out co-transport of glucose, it's basically by diffusion. It's also a direct answer. You can think of passive transport, but that's not the correct answer to this question. Diffusion actually occurs. There's a separate channel through which Cl- is moving in and moving out. And finally, in question 20, we are being asked that lipids are emulsified in the small intestine by lipase into fatty acids, and you have lipids. Lipase acts on lipids, produces two components. One are fatty acids and two are glycerol. So fatty acids plus glycerol is synthesized from lipids in the small intestine. So this basically covers all of the different questions that I wanted to discuss for today. So basically what we have discussed today includes a complete revision of the topics that were covered in interrupted for some reason, I don't know. But yes, we looked over each of those topics. We first understood the immune system. We understood antibodies, how they bind. More details are obviously on a separate course, but yes, as an introductory session, you can think of it in that way. Then we looked at further details of the immune system. We looked at how monoclonal antibodies are produced, the entire hypnoma technology, how it is executed real life. There were demonstrations for the same as well. The MENS applications of immunotechniques. Then in subsequent lectures, we understood what exactly goes into the process of sorting, cell analysis, cell sorting. And we also looked at, we also revised some parts of glycosylation, how it works, how vesicular trafficking, vesicular transport is carried out, how cell death occurs, apoptosis occurs, and basically we had like a holistic recollection of the topics that were covered. After that, we went through the assignment for this week, and finally we looked at some sample problems which are related to the topic that we have discussed, and pretty much that we will discuss, in the upcoming session for week 8. And that pretty much brings us to the end of today's session. If there are any doubts, of course, you're most welcome to post them on the discussion forum, and we'll try to get to them as much as possible. And we don't have a lot of weeks left. We will soon be looking for the exam pretty soon. So I hope that whatever doubts you have, you can come up with them and join the sessions. And without any hesitation, please mention your doubts, interrupt any instructor or any teaching assistant at any point of time, and they would be very happy to help you out in whatever ways are possible. Among my extra recommendations for today, if you want a more detailed perspective, you can go through 2B immunology, which is the best starting and I would say with the ending book for basics of immunology, for truly understanding and clearly remembering what exactly is happening in the human body. It's not even about remembering, it's about understanding. It's very well explained in that book. So for reference, you can go through that. And yeah, that is what we will wrap up with for today. In the next session, we will be taking up digestion as topic, and we'll see what all has been covered there, and you'll be submitting your assignments by then as well. So thank you for being patient, and hopefully the course is going well for you. So good night and thank you.